Okay, good morning. Welcome to Grand Rounds. I'm Jeff Miller, and with Kate Elkington and Christine Denny, I'm one of the co-directors of Grand Rounds. We're really pleased to have you join us today. I hope everyone had a nice summer. This is our inaugural Grand Rounds session for the new academic year, so welcome. A few announcements before we get started. Next week's Grand Rounds will be Wednesday, September 14th. It will be a hybrid Grand Round, so we encourage people to come to attend in person in the auditorium. It will also be simultaneously on the Zoom webinar, so you could join either way. There is socially distanced seating in the auditorium. And our speaker will be Dr. Joanna Steinglass, professor of psychiatry here at Columbia, who will be giving a talk entitled Bench to Bedside, From Mechanism to Treatment in Anorexia. And then one more event that I want to highlight and encourage you to put in your calendars and to attend is the following Monday, September 19th. There will be a welcome back kickoff reception for the academic year at 4 p.m., where Dr. Blair Simpson will give remarks in the auditorium, followed by light refreshments in the Pardis atrium in the first floor lobby right outside the auditorium. That also will be hybrid, but we want to encourage people to try to join in person if they're able to. Okay, and then just a few comments about today's Grand Rounds. I would encourage people to ask questions at any point during the talk via the Q&A feature at the bottom of the Zoom window, which is separate from the chat. And if you're a trainee, please put the word trainee at the beginning of your question. We will prioritize trainee questions. And then a plug for encouraging you to ask your question yourself. So via Zoom webinar, we're able to promote people to panelists so that we can hear you and you can ask the speaker a question yourself. You could do so with camera on or off. So if you do post a question in the Q&A, please indicate can ask question myself or prefer to have my question read, so we know what your preference is. Great, so with all of that covered, I'm really thrilled to introduce today's speaker, Dr. Anissa Abi Dargam, who I think will be known to many on the attending Grand Rounds today. She was really an outstanding colleague of ours at Columbia for many years, whose science was amazing and who really, I think, instilled a very high level of science in everyone she interacted with, in her mentees, her trainees, and her colleagues and peers. Dr. Abi Dargam is a professor of psychiatry and radiology at Stony Brook University. She received her MD degree from St. Joseph University in Beirut, Lebanon, and completed residency training in psychiatry at UT Memphis, followed by 2 research fellowships, first at NIMH and then at Yale. She's a clinical and translational neuroscientist and a leader in the field of positron emission tomography PET imaging. She's applied PET imaging in combination with multimodal MRI and clinical evaluations to study the neurobiology of severe mental illnesses, including schizophrenia, addiction, and their comorbidities. This work has uncovered alterations in dopamine transmission in specific areas of the brain in patients with schizophrenia and addiction, and also led to the generation of preclinical animal models aimed at understanding the mechanisms and consequences of these alterations, creating a truly translational program from humans to mice and back. Dr. Abi Dargam has authored over 200 peer-reviewed publications, and she's had a lot of leadership roles in our field. She's been program chair for major international meetings and served on the board of numerous scientific organizations, including the NIH Board of Scientific Counselors to the NIMH Director, Brain and Behavior Research Foundation Council, formerly NARSAD, and the Schizophrenia International Research Society. She's also been a field editor for high-impact journals, including Neuropsychopharmacology and Biological Psychiatry, and she's a member of the National Academy of Medicine, where she currently serves on the membership committee. And she's received numerous awards, including the Lieber Prize for Outstanding Research in Schizophrenia, and the Connie Lieber Research Award from the National Alliance for Mental Illness. So we're really thrilled to have Dr. Abi Dargam back at Columbia, even though it's virtual. The title of her talk today is Pervasive Disruption of Brain Functions by Dopamine in Schizophrenia. Welcome, Anissa. Thank you so much for speaking to us today. Anissa, you're muted.
Sorry.
No problem. We can hear you now.
Can you see my slides or not yet?
Yes. Do you want to just put it in slideshow mode and we'll make sure it's working? Looks great.
Okay. Okay, is everything okay? Okay. Well, thank you very much for the nice introduction, and thank you for the invitation. This very much feels like being back home. I've spent most of my career at Columbia, 20 years, the biggest stretch of my career. So it's a very special place to come back to. I would have liked to be in person, but I'll take anything. So one disadvantage of speaking at home, I realize, is that most people know most of what I'm gonna say because most of the work actually happened at Columbia. I'll give some of the updates from recent work, but the main story unfolded right there where you all are today. These are my disclosures. I wanted to explain why I chose this title, The Pervasive Disruption of Brain Functions by Dopamine Abnormalities in Schizophrenia. You know, I've been working on dopamine and schizophrenia for many decades now. And the more we understand what's going on, the more I see the disruption that the dysregulation of dopamine is creating and how it's really kind of like messing all the systems throughout the brain and leading to all types of abnormal behavior. Especially if you think about the dopamine dysregulation as being very early on in the brain, it's really interfering with normal brain development, and that's how it's leading to all these functional disturbances. So hopefully I'll give you a little sense of that today. And, you know, as usual, we start with some questions and we end up with even more questions. So I'm sorry that I will not have a lot of answers, definitive answers to almost none of the questions that we have. What are some of these questions? I think there are some fundamental questions about dopamine schizophrenia. The first one is kind of mundane, is where is the dopamine dysregulation observed? What is exactly observed? When does it emerge? And how does it emerge? And we will have some information in that regard. Another interesting fundamental question is how does one go from abnormal dopamine to symptoms and behavior? And that is really a whole field by itself. Is the dopamine dysregulation primary, leading to all kinds of consequences on other brain systems, or is it secondary, just kind of one step down the road of many other more primary abnormalities? And is it even a fair question, a possible question to ask? If it's secondary, what is it secondary to? What systems? What other system abnormalities? So this highlights how I'm gonna present the data to you today. I'll show you some of the imaging evidence that addresses some of the more mundane questions, I think. So, and that's where, you know, you may be familiar with that, but for those in the audience, especially maybe new trainees who haven't followed this type of imaging work, I will go through some of that. So I'm sorry for those who know it very well. I will present some initial studies that address the second question, and those again were done at Columbia, and the third one was mouse model. And finally, in the fourth part, I will show some of our new data that's been acquired at Stony Brook, looking at other systems that may modulate the dopamine system. So the philosophy of the work that I have implemented in my career and in my group, and that really is what is at the base of the group that I've always had, which is to bring in different types of expertise so that we can work together within the same patient acquiring the molecular imaging, which is where I got trained. I was trained as a molecular PET imager. And I think it's a very powerful technique that allows you to examine molecular targets in the brain in the living human subject. But on its own, it doesn't provide enough important information. It really has to be linked to other type of functional information, and that we acquire with sophisticated clinical assessments, behavioral assessment, cognitive evaluation, sensory processing studies, in combination with other imaging modalities such as task fMRI, resting state fMRI, MRS, and more recently, with Guillermo Jorga, implemented neuromelanin MRI, and occasionally use computational models of brain processes to be able to constrain the questions and constrain the interpretation from the data that we observed. This is on one side. On the other side, findings in humans with PET could be used to inspire or build animal models to address specific questions about mechanisms, consequences, because these could not be examined in the human brain. The purpose of all of that is really to identify targets and biomarkers to guide therapies, targets that could be addressed with treatments, and biomarkers that could be used to subdivide patients into subgroups that could benefit from one therapeutic strategy versus another, which is the point of precision medicine. So now let's just kind of start from the beginning, which is what does a patient with schizophrenia look like? And that has always struck me as just, the different types of systems that are involved in patients with schizophrenia. We have psychosis that with hallucinations, paranoia, and thought disturbance, cognitive deficits across different domains, working memory, executive functions, which is very different from social cognition that really involve different systems, different brain regions, reward deficits, mood disturbances, even movement abnormalities such as stereotypia and catatonia, and these preceded use of antipsychotics. So emotional system, sensory motor systems, cognitive systems, mood disturbance, all of that means that schizophrenia is a global brain disease. So how do you end up with a global brain disease? Could be that one alteration in one specific, very important hub affects all other regions, or it could be that there are multiple regions that are involved. One place that to start thinking about this are the Cortico basal ganglia loops. And these are, I think, critical to many conditions in psychiatry, but mostly to schizophrenia. So we have the glutamatergic projections from the cortex into the striatum, the pathway out of the striatum with the medium spiny neurons into the go, direct and no-go pathway, and the feedback from the thalamus back to the cortex and to the striatum, carrying basically the message back to the cortex as to what is the consequence of whatever input has been processed throughout these loops. The loops are divided into limbic, associative, and sensory motor, so processing all types of behavior that is relevant to humans. These systems that are so important for organizing our emotions and behavior are all very heavily modulated by dopamine. As you know, dopamine cells are housed in the midbrain, and from there project across different pathways to the cortex, as the mesocortical, subserving cognition, the ventral striatum, mesolimbic, subserving emotions and reward functions, and the nigrostriatal projections to the midsection, the associative striatum, cognitive striatum, and to the more dorsal part of the striatum, and that is the sensory motor striatum. It also, dopamine cells also project to other brain regions. So it really has a profound impact on the function of multiple regions, multiple systems. This is one reason to be interested in dopamine schizophrenia, but really the main reason I got interested is the fact, the clinical observation that all antipsychotics at the time when I started my career, all were D2 drugs. Now, unfortunately, this is still the case. We hope that there will be some exceptions soon, but up until this point, this is still the sad situation, which is that all drugs are D2 drugs. So the fortunate piece of that is that to study dopamine in human and patients with schizophrenia, we had multiple tools, and these are some of them. These are PET tools. So we, with F18 labeled F-DOPA, we can examine the activity of aromatic L-amino acid decarboxylase. So basically, the rate of transformation from DOPA or radiolabeled F18 DOPA into dopamine or radiolabeled dopamine that gets taken up in those vesicles. So the rate of accumulation of the radioactive dopamine is picked up by the PET scanner and gives you an idea of how active is AADC. That is interpreted as a measure of synthesis and synthesis capacity. We can examine the density of the vesicular monoamine transporter, the dopamine transporter, and of course, D2 receptors as well as D1 receptors. What we were interested in is to examine dopamine levels themselves, since this was one of the fundamental hypotheses of what is wrong in patients with schizophrenia. And for that, I have to show you what we've used. We basically use 3 different types of D2 scan acquisition. The first one is at baseline, where we detect a certain level of uptake of the D2 radio tracer when it binds to the D2 receptor. Some of the D2 receptors that are occupied by dopamine are not visible to the radio tracer. So we have an initial D2 binding potential. Then we change the conditions in the synapse by either flooding it with dopamine, with an amphetamine challenge, or removing dopamine. And these manipulations of dopamine will result in a different level of uptake of the D2 radio tracer. And the difference between the binding potential obtained in each condition and the baseline gives us one aspect of dopamine function. So for example, with the amphetamine challenge, we provoke a massive release of dopamine in the synapse. So the difference between the scan and the previous one is how much dopamine was stored presynaptically in the nerve terminal, while the difference between the alpha-methylparatyrosine, which is a blocker of dopamine synthesis of tyrosine hydroxylase, and we give it for 48 hours, long enough to deplete all dopamine from the synapse, actually from throughout the organism and the brain. The difference here is that those dopamine D2 receptors that were occupied by dopamine at baseline now get unmasked and are occupied by the radio tracer. So the difference between the 2 scans is the D2 occupancy by dopamine, which is related to D2 dopamine intrasynaptic levels. So storage capacity and intrasynaptic dopamine levels. Now, by using different types of D2 radio tracers, we can image different parts of the brain. And here I put in parentheses antagonists, because really the data that I will show you today is all based on use of D2 antagonist radio tracers, such as ibizam, raclopride, falipride, FLB. It may be that when you use a D2 agonist radio tracer, you have different type of information, and I did not want to get into this today because it's more complicated and we're at the initial phases of trying to understand that. But with D2 antagonist radio tracer, raclopride, low affinity, we can visualize D2 in the striatum. Falipride, medium affinity, we start to see some extra striatal sites. And with FLB, very high affinity, we can image D2 receptors in the cortex and outside of the striatum. The striatal uptake is not usable because it does not reach equilibrium during the time of the scan, which means that we cannot quantify it. So although you see this very hot blob, it's useless. What we can get with FLP is just outside of the striatum. We also subdivide the striatum to be able to examine the different functional domains. So the ventral striatum, limbic. The associative striatum was precommissural caudate and putamen and postcommissural caudate. And the sensory motor striatum was just the posterior putamen. Combining all these techniques, I'm going to show you some of those results. What has been observed is that dopamine synthesis measured with F-DOPA scans is increased in patients with schizophrenia, has also increased during the prodrome. And it predicts who will become schizophrenic, who will convert. Dopamine release is increased in the striatum in patients with schizophrenia, and intrasynaptic levels. D2 receptors are upregulated only in patients who have received antipsychotics, not at baseline. D1 receptors, DAT and VMAT2 in the striatum are normal. So there is a dysfunction with the D2 receptor, possibly function, not numbers, and with the availability of dopamine. And this data has been interpreted as excess presynaptic dopamine that is stored in those vesicles, ready to be released, and also released with regular functional activities that reflects those increased intrasynaptic levels. The effect size is around 0.7 to 0.8. This is from a meta-analysis of various techniques and various studies. This is for the striatum again. And within the striatum, the area of highest dysregulation is within the precommissural caudate, or the associative striatum as a whole, and not necessarily within the ventral striatum as we had initially suspected, or as it has been proposed. Although it is possible that some early phase of the illness could involve the ventral striatum initially, such as observed with the study in the prodrome of Raggi-Gurgis, that I will not cover here. But what we see in chronic patients is that the associative striatum is one of the main dysfunctional regions in terms of dopamine excess. In terms of what this means to symptoms, so we have found that excess dopamine relates to more psychosis, so excess dopamine in the striatum, more psychosis, and also better response of psychosis to antipsychotics. This was replicated independently by Oliver House, looking at F-DOPA in responders to antipsychotics. These are the ones that are elevated in purple, versus non-responders in blue that are similar to controls. Those do not respond to antipsychotics. So more dopamine in the striatum is almost good news, but patients with schizophrenia have at least one therapies that can help them, and that's D2 blockade. So how does one go from abnormal dopamine in the striatum to psychosis? And I was fortunate enough to be able to work with Guillermo Herga at Columbia, where we were able to combine fMRI studies during auditory task with PET imaging of dopamine and the striatum. So the idea here is that our perception, which is here schematized with this purple, peaks showing the percept, the actual experience of auditory perception. This is really a combination of two things. What we expect, the priors, which is determined a little bit by the context in which we are, and the actual stimulus. So the red and the blue together, cooked, mixed in, will give you this purple. If the prior is weak, we are more accurate in perceiving the stimulus. If the prior is very strong, this is going to have a bigger influence, and our percept is more influenced by our prior than by the sensory input. This is also illustrated in this commentary on the paper that we published. So if the context is very predictable, short stimuli are heard, a longer stimulus will be influenced by this context and will be heard as shorter. While if the context is unpredictable, uncertain, sometimes a longer stimulus, a shorter stimulus, then the target stimulus is perceived more accurately for as long as it is. This is what's called uncertainty adjustment. If the context is variable, there is an uncertainty adjustment because of this context that will basically weaken the expectation and will weaken the effects of the prior. And what we observe is that patients with schizophrenia in general have less uncertainty adjustment, and the lower the uncertainty adjustment, the more hallucinations they have. In addition, the lower uncertainty adjustment was related to more dopamine in the associative striatum and to more dopamine in the striatum as a whole. And, you know, to me, this is one of the most beautiful studies we've been able to do. And just to reiterate what we show is that the excess dopamine in the striatum led to weakening of the uncertainty adjustment. So priors were felt as stronger, which led to a more biased perception, which led to hallucinations. So these are the type of studies that we need to be able to do to understand the path from a molecular abnormality to the functional symptom. Another study we're able to do combining these techniques is the following, looking at the functional connectivity of the striatal substructures to the rest of the brain and to the cortex, where we observe that in controls, the anterior caudate is the region that is most highly connected to the rest of the brain. And this kind of pattern is lost in patients with schizophrenia. This is, again, the same region that has the excess dopamine. So one obvious, you know, potential conclusion from that, although it's a speculation, is that dopamine, the excess dopamine and the anterior caudate is affecting its capacity to have a normal functional connectivity to the rest of the brain, and that in this way is altering behavior that is processed through these cortico-basal ganglia loops. Because we also found that the more abnormal the pattern of connectivity in patients, the more psychotic they were. So just to summarize this piece, well, I've shown you that we have observed more dopamine in the striatum related to more psychosis and better treatment response, weaker altered connectivity to cortex, and more perceptual bias leading to more hallucinations. And these are just some of the papers that publish these findings. Now, moving on to the cortex, using FLB in combination with Fmri to examine working memory performance. What we observed, and this was the very first report of this observation, is that in patients with schizophrenia, on average, the group showed no displacement of the radio tracer after amphetamine, meaning on average, there was no detectable dopamine release in the cortex, unlike in healthy controls. Now, there's obviously variation with being drug naive or drug free and being older versus younger, but there is a significant group difference. And in both groups, the more dopamine release, the more activation during working memory task. What we used here is the self-ordered working memory task. We didn't find with this task a direct relationship between dopamine release and performance on the task, but we did observe it with the NBAC task, where we see that more dopamine release in patients with schizophrenia and the cortex related to better performance. So in some ways, the deficit in dopamine release, although it's a very complicated way and not easily detected with any working memory task you use, but in some way, the lack of dopamine is affecting function. Now with FLB, as I had shown you, we can detect a dopamine release everywhere in the brain. We were interested, we went into the study examine the DLPFC, dorsolateral prefrontal cortex, is what I've shown you. But what we observed is that there was a similar pattern everywhere. Outside of the striatum, patients with schizophrenia did not show much dopamine release. This is the displacement after amphetamine. They're kind of in negative, while controls are in the range that we expect. This included the midbrain, substantia nigra VTA, which is very puzzling because this is the area that houses all the dopamine cells. So, you know, we have on one hand excess in the striatum and deficit in the midbrain. So you'd think, where is the excess in the striatum coming from? If in the midbrain there's a decreased level of dopamine release that you could detect from the somatodendritic release that we can pick up there. So it is unclear how we could have this isolated excess of dopamine in the striatum. So just to, and I'm gonna go into what this observation led to, but first let me summarize. So we see that there is a dopaminergic deficit in all extrastriatal regions, including the midbrain, which led to the two following conclusions. On one hand, we reinforce the idea of cortical hypodopaminergic state associated with poor cognition, and that supports directly developing D1-based therapeutics. And I will show you the design from the study that we are currently performing in collaboration with Columbia, Yale, and UPenn, examining a D1 agonist in schizophrenia. But the second main conclusion is to try to explain how there could be a striatal excess in schizophrenia and dopamine, whether this is actually abnormal modulation of dopamine and the striatum by local mechanisms. And I'll show you some of that data. So in terms of the D1 study that I'm mentioning, you all are very familiar with this famous graph showing that and this is a kind of composite results from multiple studies from Pat Goldman-Rakik's lab and Amy Arnstein, Bita Mogadam, and others, showing that you need an optimal level of D1 stimulation by dopamine to have good working memory performance. So for example, aged monkeys or Parkinsonian monkeys will benefit, or animals will benefit from a D1 agonist, while stressed animals and amphetamine exposed animals will benefit from a D1 antagonist. This is also congruent with what we understand about the cellular functions of D1 receptors in the cortex, where we know that in layer 3 of the dorsolateral prefrontal cortex, D1 receptors on the basal dendrites are very important to the pattern of activation of these delay pyramidal cells during the delay period of the working memory, and their performance will really set the stage for the capacity for this kind of delay firing during the delay period of the working memory performance. So we were lucky to be able to work with Cerevel and these other institutions, and submitted a grant that was funded to use the Pfizer compound developed by David Gray. It's called PF064, etc. Now it's CVL562. And this is a non-catechol-based drug. The catechol-based drugs like dihydroxidine and DAR100A, which we used at one point at Columbia with Ragi Gorgas, were very difficult to use because they had very short half-life and poor blood-brain penetration, which meant we had a lot of peripheral side effects, which limited the dose we could administer and the way it could be administered. So this is a much better generation of D1 agonists. This particular drug that we are using in this study has affinity at D1 and D5, and intrinsic activity at both, although somewhat bigger at D5. There is proof of concept to support using it in schizophrenia from two types of data. The first one is in Parkinson, showing that it improves task-related. The first one is in Parkinson, showing improved motor function. And the second one is in non-human primates, showing task-related DLPFC firing improves in aged monkeys. An initial study in patients with schizophrenia did not show improvements compared to placebo. But we thought this was a drug worth revisiting. So this is the study that is currently occurring. Again, the PI is at Yale University, John Crystal and Alan Anticevic. There's a Stony Brook site, SP site with Josh Kantrowitz, SPI, UPenn, and in collaboration with Cervell, who are providing the drug. So we're using acute multiple dose design, and 90 patients with schizophrenia. They come in on 5 different days and get a different dose every day, including 1 placebo day. We're examining the effect on fMRI biomarkers during spatial working memory task. It's the working memory task that was used very consistently, initially developed by Pat Goldman-Rekiek, showing basically when non-human primates are looking at a circle and they centered in the middle of the circle and then look at the target. And it's like really after a delay, the difference between where they look and the target that shows the performance and the working memory performance. So it's a very reliable task that also has been well validated in non-human primates. So this is what we're using. The patients are on antipsychotics, but those that do not have D1 affinity to be able to basically activate the D1 receptor. We are focusing on early phase of the illness, so to avoid the kind of burnout phase, the very chronic stage of the illness. So I do not have results yet, but I can say that we've acquired almost half of the sample, if not more at this point, and hopefully within a year or so, this study will conclude and we'll have some initial results to see whether D1 therapeutics have a chance in patients with schizophrenia for cognitive enhancement. Now, the second conclusion from the FLB study was this observation that there is a deficit in all brain regions outside of the striatum and dopamine release, which led us to wonder how is the excess in the striatum emerging. And one local suspect was the cholinergic system. As you see from these loops, there are these cholinergic interneurons that are present in the striatum that modulate dopamine release in very complex ways. And so we thought if we could examine the integrity of the cholinergic system and the associative striatum or throughout the striatum, see if it's abnormal, and if it's in some way it relates to the magnitude of dopamine dysregulation, we would basically prove we would inform the question of whether the dysregulation of dopamine in the striatum is due to an abnormality in another system, that is the cholinergic system in the striatum. Now, of course, there are cholinergic terminals throughout the brain. You have the cholinergic nuclei emanating from 2 sides, the base of the brain to all the cortical projections, and then from the midbrain to the cerebellum and other midbrain areas. So we were able to examine those also at the same time as the cholinergic interneurons in the striatum. Now, the fact that the cholinergic system modulates dopamine in the striatum is just one piece that is important, but there's also the fact that the cholinergic system has been of relevance to schizophrenia for a long time. So people have been examining nicotinic agonists as cognitive enhancers with some improvement, but limited by side effects. The muscarinic system, as you may well be aware of, researchers have been examining the agonist to the muscarinic system as therapies for psychosis with xanomeline initially showing some proof of concept that now has received a lot more support in phase 2 and phase 3 trials from Karuna Pharmaceuticals, who took xanomeline and added to it a peripheral antagonist to block the peripheral side effects so that it could be administered without too much toxicity to patients with schizophrenia, and they seem to have good results on both psychosis and negative symptoms. So with that, we examined what tracer we could use to look at the cholinergic system in schizophrenia. There are multiple choices. Choices to examine receptors, nicotinic and muscarinic, were not our initial choice because they were non-selective in a way and also were not available to us, while the vesicular cholinergic transporter seemed like a good choice because at the time when we moved to Stony Brook, there was one tracer for it that seemed to behave very well. And also there is this data out there showing that the vesicular cholinergic transporter determines the levels of acetylcholine storage capacity. So basically acetylcholine availability. So we thought that would be a very powerful tool to use. And I'm going to show you the results from this study at this point. This is what the tracer looks like. And unfortunately, in terms of group differences, we do not detect any. So this is within the striatum. There's high uptake in the striatum, lower and extrastriatal regions. So these are the different striatal substructures and the striatum as a whole. Patients are in red, controls are in blue. You see no group differences. And outside of the striatum, the highest regions is the substantia innominata, globus pallidus, and then all kinds of other cortical regions, midbrain, thalamus. And the groups do not differ. Of course, it's a small sample, but at this point, there's really no difference between these groups. But we're very intrigued to see the following. When we examine a relationship between cortical vesicular cholinergic transporter and psychosis, obviously in controls there's no psychosis, so it's a flat line. But we see this relationship in patients where a higher uptake was related to more psychosis. And when we look at cognition, we see that lower cognitive performance, this is on the 2 back, is related to higher cholinergic uptake. So more cholinergic uptake was related to more psychosis and more cognitive deficit. And this latest one was not the case in controls. And controls, there was no negative correlation. If anything, it was in the direction that you would expect, more acetylcholine availability, better cognition in controls, but the opposite in patients. And this pattern of relationship to poor cognition with higher vesicular cholinergic transporter was observed through multiple cognitive domains. So at this point, this is really a kind of a preliminary observation. We did not go into the study expecting this. We were more interested in the striatum. So obviously we need to replicate it. We need to better understand it. We also need to tease apart what in that relationship is a really reliable specific signal of this tracer in the cortex. So many questions to be answered, but it's kind of an intriguing observation in the light of the data of Karuna showing a therapeutic signal with a muscarinic agonist, which makes me think that if indeed we're detecting something real here with the vesicular cholinergic transporter where there is more is associated with worse pathology, it may be necessary to look at what's happening on the postsynaptic side with the muscarinic receptor. So we would need in the next phase to combine the cholinergic transporter with a muscarinic M1 radio tracer. I don't think that there are any very specific, but that would be the next question that we will try to tackle and see if that somehow explains what we're observing here. Another suspect that we have chosen to examine in terms of regulation of dopamine is the Kappa receptor. And that this kind of emerged from a different line of evidence and interest. And this, again, started at Columbia in discussions with Sam Clark, who was very intrigued by the fact that Kappa agonists can produce psychosis, Kappa agonists like salvinorin A. And so I want to just show you some of the background that we've published in a few papers at this point. So the Kappa receptor, which is part of the opiate system, one of the receptors for the opiate system, is located throughout the brain. So it's in key regions where it could modulate dopamine. And it's been shown in preclinical studies that Kappa activation modulates dopamine release, weakens D1 versus D2 medium spiny neurons activity, modulates GABA and glutamate, so they can be relevant for multiple systems in schizophrenia. Pharmacological studies have shown that Kappa agonists can produce psychotomimetic effects, and these can be reversed by pan-opioid antagonists. Now, we proceeded to do a meta-analysis of 30 studies, including 434 patients. This was co-led by Jared Van Snellenberg and Sam Clark, examining the effects of antagonists, pan-opioid antagonists, such as naloxone, naltrexone, and nalmefene, and the mixed Kappa antagonist, mu partial agonist, buprenorphine. And this meta-analysis showed, pretty strikingly, that there is a therapeutic benefit on positive and negative symptoms, even when added to antipsychotics, and that is presumably through the Kappa antagonism, and the effect size is 0.26. And the reason I say is presumably through the Kappa system, and that's because of the following. So these are the 4 drugs. They have very low affinity at the delta, high affinity at mu and Kappa, but what they share is the antagonism at Kappa. While at mu, they have 3 of them are antagonists, 1 of them is an agonist. So obviously this cannot explain the therapeutic effect. So we think that this meta-analysis reinforces the concept of Kappa as a potential therapeutic target. So with that in mind, we proposed to do a study examining the Kappa receptor in patients with schizophrenia combined with neuromelanin studies to see if there is an increase in Kappa uptake and binding potential, and if that relates to the increase in neuromelanin uptake. Now, the study is ongoing. We've acquired to this day something like maybe 10 patients, 10 controls, and I will present this data at ACNP, but I will not present the results today because they're still under analysis. So the KOR is potentially an untapped therapeutic target in schizophrenia, and even if it doesn't show, if it's not a therapeutic benefit, it could still be potentially used for other way to classify patients according to an abnormality in specific subgroup and not in the overall population. One available drug that could be used that's an antagonist is this J&J 6795394, which is an antagonist that's being tested for anhedonia and anxiety, and currently is in trials for depression, possibly also in schizophrenia by Johnson & Johnson. So this is the beginning of the story. I don't have conclusions yet, but hopefully soon. So finally, to come to one of the last questions, is dopamine dysfunction primary or secondary? And this was addressed by the elegant work in Eric Kandel's lab, Christophe Calendonc, Eleanor Simpson's, who've continued the work in their own labs. And what this mouse was is that they created during development for a brief amount of time an upregulation in D2 receptors in the dorsal striatum to mimic a state of excess D2 stimulation that is observed in patients with schizophrenia. And what they saw, so then they turn off the gene and the mouse becomes an adult. And what they've seen as a kind of a widespread effect on circuitry throughout the brain, where the D2 overexpressing mouse, even though the alteration was very limited to the striatum, it had repercussions on the cortex, on dopamine and GABA, repercussions in the midbrain on the Q firing and NMDA expression of VTA dopamine cells, and on the structural connectivity between the go and the no-go pathway with increased bridging collaterals from the D2 into the D1 pathway. And the reason I like to highlight the study is that it gives proof of concept to the idea that if there is a very early abnormality in dopamine, it's going to lead to developmental changes that would alter circuitry throughout the brain and could basically show that dopamine could be a primary dysfunction that leads to all these other alterations. Even though I've also shown you information where we're thinking about dopamine as a result of other dysfunctions. One interesting study that supports the idea of a link or of a primary effect of dopamine that leads to a secondary effect is this relationship. It's a very small study, but it's an exciting study. It's from Gordon Frankel and Rajnarendran, published recently in Biological Psychiatry. 14 patients, 13 controls, a very small sample. But what's cool is that they showed that in patients with schizophrenia in red, high striatal dopamine was related to low cortical dopamine. They are inversely related, which means that, these in the same subjects, one could lead to another potentially, which means that we could be starting from low dopamine everywhere, and somehow there is a compensation in the striatum that leads to this excess, or the other way around, the excess in the striatum, as shown in the D2 overexpressing mouse, could lead to these abnormalities everywhere else, including dopamine and the cortex. So just to summarize this, supported by some of these later studies that I have shown you, it's possible that genetic susceptibility factors as well as environmental factors could lead to abnormal development of the dopamine system as well as other systems, leading to a neural circuit dysfunction that will translate to an abnormal regulation of presynaptic dopamine. So basically, there could be a dopamine dysregulation initially that is kind of becomes more obvious, more clinically problematic, that translate to the schizophrenia onset, as shown with the prodromal studies. But I think it doesn't stop there. There will continue to be plastic changes through the brain, especially with treatments, with antipsychotics or use of drugs that modify the dopamine system. And that kind of leads to an even different states later on in patients with schizophrenia, where there could be D2 sensitization, drug resistance, maybe lower dopamine release, and the emergence of tardive dyskinesia. So dopamine is critical to every phase of the illness and in kind of different ways. So I want to just revisit some of those questions before I end. I think the imaging evidence has given us some information about those mundane questions, but to have better information, we need to move to other systems like the induced pluripotent stem cells to examine effects of genes that are of relevance to schizophrenia risk onto the dopaminergic circuitry and its dysfunction, or possibly examine from induced pluripotent stem cells derived from patients and transformed into dopamine neurons to examine the exact cellular dysfunction that's observed, and kind of a back and forth using these systems. This is something we've been interested in, but haven't been really able to kind of implement yet. The path from dopamine dysfunction to symptoms, obviously a lot more work is needed across different functional domains, as you have seen one illustration that I have given. Is dopamine dysregulation primary or secondary? As I think more about it, I just start to wonder whether this is even a question that can be addressed, because of the fact that everything is developing simultaneously. And would we ever be able to really pin down what is primary and what's secondary when you have kind of all these synaptic or plasticity changes that are occurring in multiple systems across time? How could we go? How could we go to the most primary in a way? Is a really almost an elusive question. In terms of if it's secondary, what are the other suspects? We've examined the acetylcholine system, and more to be done here, and the Kappa system. But obviously, the more obvious suspects, some other people would say, are the glutamate system, GABA system, inflammation-mediated synaptic loss that affects dopamine system, and that could have a different effect in different brain regions. So, as I have promised you initially, we have more questions than answers at the end of these investigations. I want to conclude by acknowledging what I've shown you as work that was done by a humongous group of people. I highlight here some of the more recent work, but not to take away from the contribution of anyone, including people within the group I've had the pleasure to work with, or my collaborators, most of them at Columbia: Jonathan Javich, Eric Kandel, Jeff Lieberman, Eleanor Simpson, Christophe Calendong, and Holly Moore, who is now at NIDA, and with other sites. I want to acknowledge funding from NIMH, NIDA, BBRF. I want to thank the patients and the families who volunteered. And finally, I want to just highlight the Transcends team. This is the D1 study that's ongoing, and more recently, the Kappa study. And I want to thank you for your attention.
Wow, Anissa, that was really an inspiring talk. Thank you so much just for such a clear line of investigation that you examined it with so many different tools. And I think you did answer a lot of questions. I don't think you left more. I mean, you create new ones, but there are a lot of important findings that you shared. Thank you so much. So as a reminder, I would welcome people to put questions in the Q&A section as we move on to our Q&A part of the talk. The first question that I will ask is from Ragi Gurgis, and there's convergence because I had a very similar question in mind. So Ragi says, thank you for being such a great mentor, Anissa. My question is, given how region-specific many of the dopamine abnormalities in schizophrenia are, is there any possibility that region-specific treatments will become available or testable? If so, how long away are we from testing or using these sorts of agents? And I think the other thoughts, I was thinking along similar lines. I know there has been a literature over time about ultrasound targeted microtubule destruction, but you showed dopamine excess in striatum and dopamine deficit. It seems like if you could turn up dopamine in some parts of the brain and down in others, that might be very valuable. So yeah, we'd welcome your thoughts about that.
Yeah, I mean, first, Ragi, thank you for your nice comment. It was just a pleasure working with you, and I miss you a lot. When we have big problems here, I wish Ragi was here. So, but yeah, I mean, what you're asking is very pertinent, and I think it's one way of implementing precision medicine or precision psychiatry. Is to address the local alterations in specific ways. Now, how do you do it? How do you get to local regions in specific ways when it's something like the brain? I mean, we'd have to become very smart with pharmacology in a way. Maybe do drugs that are somehow packaged in ways so that when they're absorbed, they are only gonna go to certain sites. That is beyond my state of knowledge of sophistication of pharmacology. But I would imagine that would be something of relevance. I think Marc Caron was working on some kind of a paradigm where, it was a PDE, I don't know if it was one of those phosphodiesterase, but he had an intervention where he could address those different dopaminergic deficits in different way with just one single drug. But it may be that you have to also combine drugs. I mean, a way it may be that you have to link a D2 antagonist with a D1 agonist, or just like what the rest of field of medicine does. They do treat hypertension with one thing that could tackle the kidney, one thing that could tackle the vessels, one thing that could tackle water balance, but we don't have all these different tools. So combining those could address the regional specificity. And as you said, Jeffrey, like stimulation is an obvious one where you could target different regions, different ways. Now, in terms of the excess and the deficit and all that, I just don't know that, I kind of sometimes want to step back and saying that just dopamine is abnormal, no matter what we look at. It's very hard to have a negative finding with a dopamine study, which is weird because, with everything else, it's so easy to just, oh, we find nothing. But with dopamine, you find always something, but it's never in the, not necessarily always in the direction you're expecting. For example, D1 are up, D1 are down, dopamine is up, dopamine is down, different tracer, it's up, it's down. Maybe what we should think about is that it's abnormal. It's not doing what it's supposed to do. If dopamine, is not there in very precise ways that it should be to do what it's supposed to do, because it's a kind of almost like, this tracking different, computational tasks within the brain, and the lack of precision could lead to all kinds of abnormalities. So sometimes I want to step out of thinking it's up or down in different ways, especially when we start to think about like the D2 agonist radiotracer. They're not showing an increase in dopamine the way we would expect. They actually show the opposite. This is one of the studies that Gordon Frankel published, and our PHNO data seems to be going in the same direction, and that is in the striatum. And so it's just abnormal. And, how do we fix it? Maybe almost, another complication to the regional specificity question.
Great. And as I said, I welcome people posting questions in the Q&A. I'm happy to read them or to promote you to panelists. Anissa, another thing that I thought was really interesting that I noted with the cholinergic finding that you shared, the newer data, was that you didn't find case control differences, but you found correlations within the patient group, right? Positive correlations with symptom severity of psychotic symptoms and negative correlations with cognition. And in our PET work, that's something that we've been seeing a lot of recently, is that in depression, with respect to depressed groups, suicidality, we often are observing correlations within a patient group with symptom domains and severity, even when we don't find case control differences. And I'm curious sort of how you think about that and how you use this tool and study design, like how you move forward our understanding of pathophysiology. How important is it to find healthy volunteer versus patient group differences versus looking for these correlations with the symptom domain?
Yeah. Well, I think when you find group differences, it makes your life easier. Publishing is easier, a grant is easier. But I mean, how it's difficult to find group differences when you think with pets, especially how small the samples are and how heterogeneous the population is. So to find a group difference is almost miraculous. And I think that it's really striking that the dopamine studies keep coming up with group differences. So I'm not disturbed by the fact that there is no group difference, but that there is a relationship to a specific functional system. Because, how could it be that you just take 10 patients, 10 controls, and suddenly, they're going to be abnormal? And the brain is so complicated in some ways. In a way, I think that we've been kind of expecting group differences, but the more I see data and the more I become old, I think that we need to tried to think about the data critically and tried to think what is it telling us, and then design studies that follow up on these observations and not dismiss them just because there are no group differences.
Great, thank you. We have a few questions that have come in, additional questions. The next one is from Deanna Martinez, who says, hello, Anissa, glad to hear about your progress on this line of thought. Can you address this question? Could existing medications that block Kappa receptors, such as naltrexone, be used now to augment treatment in schizophrenia?
Yes, so there have been studies. I think that naltrexone may have been one of them, but you know, it is possible that this could be used now to just in a kind of a nice, well designed double-blind study to look at, because the effect size we see was a meta-analysis 0.26 with patients already treated, so they've already had the benefit of the antipsychotics. So I think that that's possible. I haven't really thought of it because I guess I'm not in the mode of thinking, oh, let's do a clinical trial. I find them too slow for me. And also maybe the funding agencies don't like me doing clinical trials. For some reason, we proposed a clinical trial not too long ago and said, no, do a mechanistic study, take out the clinical part. So, okay. So yeah, Diana, they won't trust me with that, but I think that is a good suggestion.
That's very funny. Okay, our next question is from Christine Denny, who asked if you could comment on what mouse models you think are the best to utilize to study endophenotypes or end phenotypes of schizophrenia. She noted that you mentioned Eleanor and Chris Kellendonk's work, and do you see other models on the horizon? If you could comment on that.
Yeah, I mean, there are lots of models, and I may not be the best person to answer this question, but one obvious one is at Columbia that we call the GOGOS model, right? The 22q11 deletion model. This is a very interesting mouse because it's based on, you know, one of those risk factors that has a big effect on the risk for schizophrenia. So, and this mouse has led to some very interesting observation. One of them that I probably can mention is that in one of the mutations that is part of the 22q11 deletion, or one of the deleted genes, that the mouse was observed to have increased dopamine cell firing in the area that projects to the sensory, to the associative striatum. So that is very interesting. As an abstract that will be at the SFN meeting this year. There may be other findings that I'm not aware of, but definitely this mouse is really relevant. The inflammation type of mice are also important because of the role of inflammation, you know, and the genetic findings with the immune system involvement in schizophrenia. You know, other types of mouse model would be based on specific risk genes that one can use to create a mouse model and see the effect of that gene on the dopaminergic system and the rest of the, you know, relevant systems and in the mouse. I think what's important with all these models is that it's kind of an interactive kind of conversation. This is why I liked working with basic scientists and we had these very fun programs, the Conti centers when I was at Columbia, because, you know, you have to interpret what you see in humans, feed into the animals, interpret the data in animals. For example, what the finding of Christophe and Eleanor was the bridging collaterals. So structural changes within the basal ganglia, that is currently being tested. The study is completed, the analysis is pending, tested in patients with schizophrenia to see if there are altered connectivities the same way that was described in the mouse. You know, other mouse model, I mean, I think that the MAM model has been also very relevant. This is kind of a general toxicity on the cortex. So, and that has a big hit on the hippocampus. And this mouse model that was developed by Holly Moore and Tony Grace shows abnormalities in dopamine regulation via the altered hippocampal drive onto the dopamine system. So models are useful because they give information about the different systems that are tackled within each one of those.
Great, thank you. We have a few more questions. The next one is from Mike Grunbaum, who asks if you could comment on how clozapine fits into the dopamine picture that you have described so eloquently.
Well, I mean, it's still a D2 blocker. So, I think that so when you give 300 milligrams of clozapine, you're getting, somewhere less than 60% occupancy. When you push it all the way, you may get to 60% occupancy, but we don't know how much occupancy of the D2 receptor is necessary to get a therapeutic effect. You're still getting a substantial effect. Clozapine is used at doses that lead to D2 occupancy. So that is still part of the picture. But what else is it doing that makes it superior? I think is the important question. And, I don't think that we have a good answer to that at this point. Almost every target that clozapine has been taken in isolation and looked upon, unless there are targets we are not seeing yet that haven't been tested. We still don't know.
Got it. Thank you. The next question is from Guillermo Horga, who's having issues with his connection, so prefers for me to ask this. He says, thanks for your inspiring work and mentorship, Anissa. It was great hearing about your more recent work. I had a general question about the need for more studies on dopamine and schizophrenia. Some people, including grant reviewers, tend to think of this area as not novel and one that is less likely to produce newer treatment strategies. In contrast, you showed that many important questions are unanswered. Any thoughts on this?
Yeah, I mean, this is a very important question. I think in this field, we tend to never want to answer anything conclusively. It's like we just move on and we get into, we're almost ADHD.
Right.
We have a lack of dopamine as a field. So I am of the school that if a study is well done and is going to answer an important question, that it should be done, because we never dismiss things completely. I mean, we know, can anyone in their right mind think that dopamine is not important to schizophrenia? It's impossible. But do we know exactly how, and have we pushed it to the limit of where we need to go? No. And, unfortunately, the system is, I don't know, the system is imperfect, and it's very difficult. And I've sat on study sections where I've had these fights, whether it was about dopamine or something else, where, it's kind of a reflection of who's on the study section at that point in time, and they are going to determine what gets done. There isn't like this superior intelligence that is deciding what to do. But it's a little bit like, I think you may have had, or we had a similar discussion about biomarkers. You have all these candidates, but nothing is pushed to like, okay, is it yes or no? Can we just drop it or move it forward? We just don't do that. So, oh, this is another candidate biomarker. Isn't that nice? Let's move on to another one that's novel, so that the reviewers will give us novelty.
Right. And then Guillermo drilled down with a more specific question. He said, you talked about the possibility that dopamine is modulated locally in the striatum. Do you think the primary issue could be at the level of specific SNVTA neurons, or modulation at the midbrain level, and somehow producing alterations in different cells, leading to the striatal cortical correlation in the Frankel study?
Yes, I think both are on the table. We haven't excluded either hypothesis. It could be that a very specific pathway, I mean, dopaminergic cells and the midbrain are very diverse, and they don't like behave the same way and across the range, and they also are not possibly modulated the same way. And we need a lot more specific information. So is it possible that this a very small subset of cells projecting exactly to that brain region, which is in the rostral caudate, is acting differently, or has a specific abnormality that is then leading to all these consequences? Or is it just everything is kind of pretty much the same, but in the striatum, there's a specific regional abnormality with just the terminal release? Unfortunately, we don't have the tools to answer these questions yet, but I think both are possible at this point.
Great. Thank you. The next question comes from Talal, who says, great presentation, Dr. Abi Dargam. Are there data that depict dopaminergic abnormalities that are specific to treatment-resistant schizophrenia?
Well, so that would be what I showed in a way, the FDOPA study of Oliver's House showing the responders versus non-responders. So the non-responders are low on FDOPA. And my own study, which preceded the FDOPA study, which showed that people who had low the dopamine release did not respond to medications after 6 weeks of inpatient treatment. So I think that we can conclude from that treatment resistance means normal dopamine levels, which means, they have some other abnormalities that's leading to their psychosis. Could it be the cholinergic system, like what we saw, abnormal muscarinic cholinergic situation and the cortex, or is it the glutamate? Or, we don't have enough information about the other systems that are at play in the non-dopaminergic patients.
That actually makes me curious. I was wondering, as you're presenting that cholinergic work, whether you are planning to look at the cholinergic system and the dopamine system in the same patients to try to get at some of those questions about their interrelationship or independence.
Yeah. So, I mean, yes, absolutely, that would be great. The problem is what Guillermo alluded to, which is any study you propose that has extensive dopamine imaging is going to be felt as, oh, again, no, not again. So I think that the solution to that we recently have been adopting is really thanks to Guillermo, is to try throw in a neuromelanin MRI scan. They're less upset about that because it's more novel.
Got it.
So, I mean, you don't really get the cellular specificity and the regional specificity as well, but it's still a good proxy, hopefully. It's being currently validated, and hopefully with time we'll have more confidence in what it's telling us. But yeah, I think it would be nice to always do that in combination, see whether they go hand in hand or whether they're in opposite different groups of patients.
Yes, great. And then I think this will probably be our final question for today. It's from Eleanor Simpson, who says, wonderful talk. Are you continuing to study dual diagnosis patients? Might they provide more information about changes in other non-dopamine systems, opioid, acetylcholine, etc.?
Yeah, thank you, Eleanor. It's great to hear from you. Yes, so actually I was very lucky when I moved to Stony Brook that Scott Muller got interested in, for geographical reasons, to move to Long Island, and I was able to recruit him, and we've been having great collaborations and dual diagnosis patients. So we have studies looking at the we've done a few studies, but we plan a lot more. And in particular, now we're going in the direction of insight, because he is an expert on insight. We're examining insight in patients with schizophrenia, addiction, and comorbid patients. We're also looking at the cholinergic system, and maybe we'll do more. But yeah, this remains a very important area that we want to develop further, because patients with schizophrenia are prone to all kinds of comorbidities, and drug addiction is a big one.
Great. Well, that was truly a fascinating talk and discussion. It was really so interesting to hear, Anissa, where your work has gone since you were with us at Columbia. We miss you, and thank you for an amazing talk and spending time with us today.
Thank you very much. Bye.
Bye.